Today, we're going to explore one of the most transformative innovations in modern science, a technology that has the potential to redefine how we understand and treat diseases, how we approach genetic research, and even how we might address global challenges such as food security and climate change. This technology is known as CRISPR, and it's not just a tool but a revolution in the making. To truly grasp the magnitude of CRISPR, let's begin by understanding where it comes from. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, a term that might sound complex, but at its core, it refers to a naturally occurring defense mechanism in bacteria. These microorganisms have evolved over millions of years to survive in environments where they are constantly under attack from viruses. When a virus invades a bacterium, the bacterium has a unique ability to capture and store snippets of the viral DNA in its own genome. These snippets are arranged in a very specific pattern, which is where the name CRISPR comes from. Now, what's truly fascinating about this process is that the bacterium uses these stored snippets as a kind of genetic memory. The next time the virus tries to invade, the bacterium can recognize it by matching the viral DNA to the stored snippets. It then deploys a molecular tool to cut up the viral DNA, effectively neutralizing the threat. This tool is the Cas9 enzyme, which acts like a pair of molecular scissors. It's this natural system that scientists have harnessed to develop CRISPR technology for gene editing. CRISPR has two main components, the Cas9 enzyme and a guide RNA. The guide RNA is a short strand of RNA that is designed to match a specific sequence of DNA in the genome. When the guide RNA binds to its target sequence, it directs the Cas9 enzyme to that exact spot, where the enzyme can then make a precise cut in the DNA. Once the DNA is cut, the cell's natural repair mechanisms kick in. These mechanisms can either repair the cut in a way that disables the gene or allows scientists to introduce new genetic material at the site of the cut. To put this in perspective, imagine you are editing a massive book, where each chapter represents a chromosome and each sentence is a gene. Traditional methods of genetic modification were like trying to rewrite a sentence with a typewriter. You could add or delete text, but it was difficult to make precise changes. CRISPR, on the other hand, is like using a modern word processor with a find and replace function. You can locate a specific word or phrase and make exact changes without affecting the rest of the text. The precision of CRISPR is what makes it so revolutionary. This technology allows us to edit genes with an accuracy that was previously unimaginable. We can now target specific genes and make precise modifications, opening up possibilities for treating genetic disorders that were once considered incurable. For example, sickle cell anemia, a debilitating genetic disorder, is caused by a single mutation in the hemoglobin gene. With CRISPR, scientists can target and correct this mutation, potentially curing the disease at its genetic root. But CRISPR's applications extend far beyond curing genetic disorders. In the field of oncology, for instance, CRISPR is being used to develop new cancer treatments. One promising approach is CAR T-cell therapy, where a patient's own immune cells are genetically engineered to better recognize and attack cancer cells. Here's how it works. T cells, which are a type of white blood cell, are extracted from the patient's blood. These cells are then modified using CRISPR to express a receptor that targets cancer cells. Once these engineered T cells are infused back into the patient's body, they can seek out and destroy cancer cells more effectively. This approach has shown remarkable success in treating certain types of blood cancers, such as leukemia and lymphoma, and researchers are now exploring its potential for other forms of cancer. Beyond human health, CRISPR is also being used to address challenges in agriculture. The global population is expected to reach nearly 10 billion by 2050, and this will place immense pressure on our food systems. CRISPR offers a way to create crops that are more resilient to environmental stresses, such as drought, heat, and pests. For example, rice, a staple food for billions of people, is highly susceptible to bacterial blight, a disease that can devastate yields. Scientists have used CRISPR to develop rice varieties that are resistant to this disease, which could help secure food supplies in regions that are particularly vulnerable to climate change. Moreover, CRISPR can be used to enhance the nutritional content of crops. For instance, researchers are working on increasing the levels of essential vitamins and minerals in staple foods like wheat and maize. This could have a profound impact on global health, particularly in developing countries where nutrient deficiencies are common. By fortifying crops with higher levels of iron, zinc, and vitamin A, CRISPR could help reduce the incidence of malnutrition and related health issues. However, as with any powerful technology, CRISPR also raises significant ethical and societal questions. The ability to edit the human genome has profound implications for what it means to be human. One of the most contentious issues is the possibility of making heritable changes to the human genome, alterations that could be passed on to future generations. This raises the specter of designer babies, where genetic modifications are made not just for medical reasons but for enhancing physical, cognitive, or even aesthetic traits. Such possibilities bring up questions about social inequality, access to genetic technologies, and the potential for new forms of discrimination. There is also the issue of unintended consequences. While CRISPR is highly precise, it is not infallible. Off-target effects, where the Cas9 enzyme cuts DNA at unintended locations, remain a concern. These unintended cuts could potentially disrupt other important genes, leading to unforeseen health problems. As such, scientists are working to improve the accuracy of CRISPR, developing new versions of the technology that can minimize these risks. For example, base editors and prime editors are newer tools that allow for even more precise modifications without making double-strand breaks in the DNA, which can reduce the likelihood of off-target effects. Another important aspect of CRISPR research is the delivery of the CRISPR components into the target cells or tissues. Effective delivery is crucial for the success of gene-editing therapies, especially when targeting diseases that affect multiple organs or systems. Various delivery methods are being explored, including viral vectors, nanoparticles, and direct injection. Each method has its own advantages and challenges, and ongoing research is focused on optimizing these delivery systems to improve the efficacy and safety of CRISPR-based therapies. Beyond its medical and agricultural applications, CRISPR is also revolutionizing the field of evolutionary biology. By enabling precise genetic modifications in a wide range of organisms, CRISPR allows scientists to explore the genetic basis of evolution and adaptation in ways that were previously impossible. For example, researchers are using CRISPR to study how specific genes contribute to the development of complex traits, such as behavior, physiology, and morphology, in various species. By editing or knocking out these genes, scientists can observe the resulting changes and gain insights into their evolutionary significance. This research is shedding light on the genetic mechanisms that drive evolution and the diversity of life on Earth. In addition, CRISPR is being used to investigate the potential for de-extinction, the revival of extinct species through genetic engineering. This is a concept that was once purely speculative but is now being seriously considered as a possibility. By editing the genomes of closely related living species, scientists are exploring the potential to bring back species like the woolly mammoth or the passenger pigeon. While this research is still in its early stages, it raises intriguing questions about the limits of genetic engineering, the ethical considerations of reviving extinct species, and the impact such actions could have on current ecosystems. The future of CRISPR holds endless possibilities. As our understanding of genetics and molecular biology continues to advance, CRISPR will undoubtedly play a central role in shaping the future of science, medicine, and agriculture. The ongoing research into improving the precision, efficiency, and safety of CRISPR is opening up new avenues for its application, and the potential impact on human health, food security, and environmental sustainability is immense. In the years to come, we can expect to see CRISPR being used to tackle some of the most pressing challenges facing humanity. For instance, antibiotic resistance is a growing global health threat, and CRISPR could be used to develop new treatments that target resistant bacteria. Environmental conservation is another area where CRISPR could make a significant impact, such as by creating genetically modified organisms that can help clean up pollutants or restore damaged ecosystems. Moreover, CRISPR is likely to become a standard tool in biotechnology and synthetic biology, enabling the creation of new organisms with tailored functions and characteristics. This could lead to breakthroughs in biofuels, bioremediation, and sustainable manufacturing, transforming industries and economies worldwide. However, with great power comes great responsibility. As CRISPR technology continues to evolve, it is essential that we navigate its ethical and societal implications with care and foresight. The choices we make today will shape the future of life on Earth, and it is our duty to ensure that this powerful tool is used for the benefit of all. One of the most critical areas of ongoing research is understanding and mitigating the long-term effects of gene editing, particularly in humans. While the potential for curing genetic diseases is immense, we must also consider the risks of unintended genetic consequences that could arise from CRISPR edits. Scientists are still learning about the complex interplay of genes and how changes in one part of the genome can affect others. There is also the question of how edited genes might interact with the environment, lifestyle, and other factors over a person's lifetime. As such, long-term studies and careful monitoring will be essential to ensure that CRISPR-based therapies are both effective and safe. Another consideration is the regulatory landscape for CRISPR and gene editing technologies. Different countries have different regulations and guidelines regarding the use of CRISPR, particularly when it comes to editing the human genome. In some regions, there are strict prohibitions against germline editing, where changes are made to eggs, sperm, or embryos, because these edits can be passed on to future generations. In other places, there is more flexibility, allowing for research and potential clinical applications under certain conditions. As CRISPR technology advances, international cooperation and dialogue will be crucial to establishing ethical guidelines and regulatory frameworks that protect individuals and society while allowing for scientific innovation. The public perception of CRISPR and gene editing is also a key factor in its future development and application. Public understanding and acceptance of gene editing technologies will influence how they are implemented and regulated. Education and transparent communication about the science, benefits, and risks of CRISPR are essential to build public trust and ensure informed decision-making. Scientists, ethicists, policymakers, and the public must work together to navigate the complexities of gene editing in a way that maximizes benefits while minimizing risks. As we look to the future, it's clear that CRISPR has the potential to address some of the most significant challenges facing our world. Whether it's curing diseases, feeding a growing population, protecting our environment, or understanding the fundamental processes of life, CRISPR is poised to play a central role in shaping the future of humanity. However, the path forward will require careful consideration of the ethical, social, and scientific implications of gene editing. By approaching CRISPR with caution, responsibility, and a commitment to the greater good, we can harness this powerful technology to create a better future for all. We've talked about how CRISPR has the potential to revolutionize medicine, agriculture, and even evolutionary biology, but there's still so much more to explore about the broader implications of this technology. The ripple effects of CRISPR extend into areas such as bioethics, global health equity, environmental conservation, and even the philosophical questions about the nature of life and identity. One of the most critical aspects of CRISPR's development is the conversation surrounding bioethics. The ability to alter the genetic code of living organisms, including humans, is a power that comes with significant moral and ethical responsibilities. When it comes to editing the human genome, particularly in ways that affect future generations, we must tread carefully. The debate over germline editing, which involves making genetic changes that can be inherited, is one of the most contentious issues in the field of bioethics. On one hand, the potential benefits of germline editing are enormous. Imagine a world where we could eliminate inherited genetic disorders, such as cystic fibrosis, Huntington's disease, or muscular dystrophy. These are conditions that cause immense suffering for millions of people around the world, and the idea of being able to prevent them at the genetic level is incredibly compelling. However, the flip side of this is the risk of unintended consequences. Our understanding of genetics is still evolving, and while CRISPR allows us to make precise edits, there's still much we don't know about how these changes might affect other parts of the genome or how they could interact with environmental factors. Moreover, there's the possibility of creating new genetic problems that we didn't anticipate. Then there's the issue of access and equity. Who gets to decide who has access to these powerful technologies? Will CRISPR be available only to the wealthy and privileged, or will it be accessible to everyone, regardless of their socioeconomic status? This raises important questions about justice and fairness. If gene editing technologies become widespread, we could see a future where genetic enhancements create new forms of inequality. Those who can afford to edit their genes or the genes of their children might have advantages over those who cannot. This could exacerbate existing social inequalities and create new forms of discrimination. Furthermore, the international community faces the challenge of regulating the use of CRISPR in a way that respects cultural differences while also protecting human rights. Different countries have different views on the use of genetic technologies, particularly when it comes to altering the human germline. For example, some countries have banned germline editing altogether, while others allow it under certain conditions. As CRISPR technology becomes more advanced and more widely available, it will be crucial to develop international guidelines and agreements to ensure that it is used responsibly and ethically. The environmental implications of CRISPR are another area that warrants careful consideration. In addition to its potential applications in agriculture, CRISPR could be used to address some of the most pressing environmental challenges we face today. For example, scientists are exploring the possibility of using CRISPR to create genetically modified organisms that can help combat climate change. This could involve editing the genes of plants to enhance their ability to absorb carbon dioxide from the atmosphere or modifying microorganisms to break down pollutants in the environment. While these applications hold great promise, they also raise important questions about the potential risks and unintended consequences of releasing genetically modified organisms into the wild. There's also the potential for CRISPR to be used in conservation efforts. For example, scientists are exploring the use of CRISPR to help save endangered species by editing the genes of animals to make them more resilient to disease or environmental changes. In some cases, this could involve reintroducing genetic diversity into populations that have been reduced to small numbers due to habitat loss or other factors. However, these interventions must be approached with caution, as altering the genes of wild populations could have unpredictable effects on ecosystems. The philosophical implications of CRISPR also deserve consideration. The ability to edit the genetic code of living organisms challenges our traditional notions of identity, individuality, and what it means to be human. If we can alter our genes, are we still the same person we were before the edit? How do we define identity in a world where our genetic makeup can be changed? These questions are not just academic, they have real-world implications for how we understand ourselves and our place in the world. Another interesting area of research is the potential for CRISPR to be used in synthetic biology, where scientists create new forms of life with specific functions. This could involve designing microorganisms that can produce biofuels, clean up environmental pollutants, or even create new materials. The possibilities are vast, but they also raise questions about the ethical implications of creating new forms of life. What responsibilities do we have to these organisms? How do we ensure that they are used for the benefit of humanity and the environment, rather than for harmful purposes? The future of CRISPR also includes its potential integration with other emerging technologies, such as artificial intelligence, AI, and big data. These technologies could be used to enhance the precision and efficiency of CRISPR by analyzing vast amounts of genetic data to identify the best targets for gene editing. AI could also help predict the outcomes of genetic edits, reducing the risk of unintended consequences. However, the integration of these technologies also raises concerns about data privacy, security, and the potential for misuse. As we move forward, it's clear that CRISPR has the potential to change the world in profound ways. But with this power comes the need for careful, thoughtful consideration of the ethical, social, and environmental implications of gene editing. It's up to all of us, scientists, policymakers, and the public, to ensure that CRISPR is used responsibly and for the benefit of all. The journey of CRISPR is just beginning, and we are only starting to scratch the surface of what is possible with this technology. As research continues, we can expect to see even more exciting developments in the coming years. Whether it's curing diseases, enhancing human health, protecting the environment, or exploring new frontiers in synthetic biology, CRISPR is poised to play a central role in shaping the future of science and society. However, it's important to remember that with great power comes great responsibility. As we unlock the potential of CRISPR, we must also be mindful of the ethical and social challenges that come with it. By working together and approaching this technology with caution and foresight, we can ensure that CRISPR is used in a way that benefits all of humanity. In conclusion, CRISPR represents a new era in genetic science, one that offers unprecedented opportunities to improve human health, enhance food security, and address environmental challenges. But it also requires us to grapple with complex ethical questions and navigate uncharted territory in terms of regulation and public perception. As we continue to explore the potential of CRISPR, it's essential that we do so with a commitment to using this technology for the greater good, ensuring that its benefits are shared by all and that its risks are carefully managed. The future of CRISPR is bright, but it's up to us to ensure that it's a future we can all embrace. As we wrap up this deep dive into the science behind CRISPR and its incredible potential to revolutionize everything from medicine to agriculture, I want to thank you all for joining me on this journey. We've explored the fascinating origins of CRISPR, its groundbreaking applications, the ethical considerations, and the profound impact it could have on our future. CRISPR is more than just a tool, it's a doorway to a future where we can solve some of humanity's biggest challenges. If you found this video informative and thought-provoking, please give it a thumbs up. Your support helps me continue creating content like this. Don't forget to hit that subscribe button and ring the notification bell so you never miss an update. I post videos regularly on the latest in science, technology, and how these advancements are shaping our world. Also, I'd love to hear your thoughts on CRISPR. Do you think the potential benefits outweigh the risks? What ethical concerns do you think we should be most mindful of? Let me know in the comments below. I read every single one and look forward to hearing what you have to say. And if you're interested in learning more or diving deeper into topics like this, be sure to check out the links in the description for further reading and some of my other videos on related topics. Thank you again for watching, and until next time, keep exploring the wonders of science. Stay curious, stay informed, and I'll see you in the next video.